Okay, I'm Stefano Monti, faculty in the section of computational biomedicine. So you heard from Tom, Stacy, and George. Let me connect the threads from the point of view from the lens of molecular epidemiology and in silico biology. So here is in a nutshell and highly simplified how most of our aging studies are designed. Basically, we start with sample collection, biospecimens, blood and stool samples from individual spanning a wide range of ages, including long-lived individuals, centenarians, supercentenarians. Then next, basically, we extract key molecular entities, genes, transcript, proteins, metabolites, microbes from plasma and stool samples of age-stratified individuals that are actually annotated with multiple clinical traits of phenotype, including the cognitive deep phenotyping that Stacy has alluded to. And basically, so this goes to fill in massive data set, genes or molecular feature by subjects. And basically, next what comes is basically in silico biology, which is about connecting the rows of those big matrices, the molecular entities, with the columns, which are the clinical phenotypes. And so that's where actually data science, a combination of computer science, statistics, and machine learning comes in, to try to basically understand and study, basically identify the molecular factors that contribute to healthy aging and longevity. So as Tom pointed out, we're involved in multiple consortia that actually all more or less take this experimental design with variation, of course. So Tom mentioned the Long Life Family Study, the Longevity Consortium, the Integrated Longevity Omics. They all have slightly different aims, but they have this the common mission, which is basically going after the factors that contribute to healthy aging and longevity. What extends the so-called health span as distinguished by the lifespan. And so here are some examples, and these are all studies that basically include pretty much everybody who spoke before me. For example, we performed a single-cell transcriptomic studies in centenarians, which pointed actually to a better immune system in centenarians compared to non-centenarians. Another study of bulk proteomics of a cohort from the New England centenarian study showed that basically the centenarians had a slower aging rate. And actually the most recent study of metabolomics, bulk metabolomics, identifies metabolite signatures of aging, longevity, and survival. And these are just kind of a few samples of multiple studies that have been completed or are ongoing, and all aimed at identifying these factors that contribute to healthy aging and longevity. But let me actually wrap it up by actually pointing to one of the main challenges that is in the centenarian studies. When you compare centenarians to non-centenarians, it's very hard to tell what is actually a marker of extreme longevity, of exceptional longevity versus a marker of end of life or extreme old age. This is different. It's difficult to distinguish the aging factor from the resilience factors. And so basically this complicates the experimental design, and the approaches that we take are, for example, investigating offspring of centenarians, which of course are likely to harbor some genetic factors that contribute to their longevity, as well as actually a longitudinal study where basically we follow and profile subjects over time, and so we can identify age trajectories. So this all points basically to how difficult aging studies are, and I will stop here.